guys, my name is Priscilla Criasso and I'm with
Stephanie Calvo.
And today we will be doing a presentation on a research paper titled Y Chromosome Sequences in Turner Syndrome, Multiplex PCR, a New Method for Diagnosis. This research paper will discuss what Turner Syndrome is, as well as a new method to identify the Y chromosome fragments that can be found in individuals with Turner Syndrome, as opposed to the old method, which was known as karyotypes. So, to begin, the introduction of this paper will discuss what Turner Syndrome is. Turner Syndrome is a disease that occurs in 1 in every 2,500 females. It is basically where one of the X chromosome is either partially missing or completely absent in the cells of a female. Even individuals with Turner Syndrome experience symptoms such as sexual infantilism, short and winged neck, cubitus valgus, which is basically where the elbows are fused and pointing outward, a short stature, and gonadal dysgenesis. The problem with individuals with Turner Syndrome is that their gonadal dysgenesis, if there is a Y chromosome present in their cells, the material in the Y chromosome will actually cause gonadal tumors because of their gonadal dysgenesis. So it is important that these Y chromosome fragments are actually found. So normally the method used is karyotypes. A karyotype is basically a visual representation of all the chromosomes inside of an individual's cells. So if you look at the bottom photo, it basically shows you what a karyotype is and how an individual with Turner Syndrome only has one X chromosome. However, karyotypes are not able to show you all the time the Y chromosome fragments that are found in an individual's cells with Turner Syndrome. So these researchers have discovered a new method to identify these Y chromosome fragments, and this is known as multiplex PCR. Multiplex PCR is different from normal PCR because in traditional PCR, you basically get different samples of the gene and you have to place them in different tubes and do it one by one. However, in multiplex PCR, you get every single sample of the genes that you want to amplify and along with their primers, and you stick them in one tube and you then put them through gel electrophoresis. The methods of this experiment will be discussed by Stephanie Calvo.
Okay, so hi guys. So for today, the methods of the research paper include 109 patients and 2 controls. The 2 controls is a healthy female with 46 chromosomes, while the healthy male has 46 chromosomes. So 109 blood samples and 2 blood samples of the healthy were included in the MPCR, 3 milliliters. So primers were made for the multiplex PCR, 1 on the X chromosome, which is MLX, and 2 on the Y chromosome, SRY gene, and TSPY. The MPCR goes through thermocycling, which is 3 different stages: denaturation, annealing, and extension. So in the first part, in denaturation, it is at 94 degrees Celsius. The DNA opens up so that the primer could eventually attach, which happens in annealing, which is 50 to 60 degrees Celsius. The primer for that specific gene attaches to the gene sequence, and the DNA polymerase is then able to complete the copy, which happens in the third step, extension, 72 degrees Celsius. The DNA polymerase copies and completes the DNA sequence that the primer binded to. Once this MPCR is complete, then the DNA is put into gel electrophoresis in order to be analyzed and compared to the 2 controls. So, Table 1 describes the primers made for the SRY gene, the TPSY gene, and the MLX gene. So, this shows you the forward and reverse gene sequences where the primer will attach in order for the DNA polymerase to copy the DNA sequence. And it also introduces how many base pairs are in each gene. So, this gives a visual representation of the multiplex PCR, which contains 3 different strains, or in our case, 109 different strains. And they are put into one tube into the MPCR and then eventually put into the gel electrolysis for analysis.
Results and discussion of this paper. 14 out of the 100 patients, which is 12.8%, were found to test positive for the fragments of the Y chromosome. This was through the MPCR. This was more effective than the normal karyotyping, which was commonly used before that, which only detected 9.9% of the Y chromosome fragments. However, there were difficulties discussed in using MPCR. One of the difficulties was adjusting the amplification in the standardization of MPCR because of the low intensity of the SRY and AMyloX genes in the agarose gel in electrophoresis. However, this difficulty was overcome by changing the DNA polymerase as well as increasing the magnesium concentrations in the primers used. So there are also benefits that were discussed for the use of MPCR. The benefits included that it was economical, less time, more information, and greater precision. It was more economical because you used less reagents and materials in order to carry out the experiment. It also included less time. You used less reagents per unit of time.
I'll cut out that part.
The experiment, because of the fact that you used less reagents, there was less time taken in completing the experiment. You also got more information per unit of time, and there was greater precision and less error because less reagents were used and materials. In this image that was placed in the discussion section of this paper, it shows you a graph of the two controls, the healthy female and the healthy male. It shows you the absorbance of the 3 genes, or the 1 gene being looked at, versus the time that it took for them to travel up the electrophoresis gel. In the male, you can see that the longer the base pair for the genes, that the longer time it took for them to travel up the electrophoresis gel. The AMyloX gene therefore traveled up. It took the longest time for the AMyloX gene to travel up the gel, while the SRY gene, which had the shortest number of base pairs, took the least amount of time. And the absorbance was the most for the TSPY gene in the male. This is a photo of the electrophoresis gel that occurred in this experiment. Basically, it shows you the 2 controls, the female and their 1 AMyloX gene, which traveled up the least amount relative to all the other ones. In the male, it shows that the male had all 3 of the genes, the AMyloX, TSPY, and the SRY gene. The SRY gene did travel up the furthest. And in one of the patients with Turner syndrome, you can see that they did present fragments of the TSPY and SRY gene, as well as the AMyloX gene.
So in conclusion, it could be determined that MPCR is a better technique for the detection of the hidden Y, as the gel electrophoresis indicates that a patient could, compared to a healthy male individual, have the same genes exhibited, while karyotype cannot show the hidden Y component. And it is also determined that MPCR is a practical approach with minimalization of errors. Thank you.